Good afternoon, and thank you for joining us for Webinar Wednesdays with PKD Foundation Chief Scientific Officer, Dr. David Barron. I'm Alexis Denny, and I work in the Research and Education Department here at the PKD Foundation. I will be with you throughout the talk, so if you have any trouble, you can send me a message via your chat box for help. We're also live tweeting throughout the event. Be sure to follow us at PKD Foundation and tweet questions using the hashtag Webinar Wednesday. We'll add your questions to the queue to be answered at the end. Today, David will be talking about the genetics of PKD. During the presentation, please type your comments and questions into the chat panel on the right side of your screen or tweet us, as I mentioned before. We will have a Q&A at the end of the session to answer as many questions as before. Please note that the PKD Foundation does not offer medical advice, and the information shared this afternoon is not intended to be a substitute for consultations with your healthcare professional. Care and treatment decisions related to your health must be made in consultation with your health professional team. We are recording this webinar and will archive the recording on our website within the next few days. And now I'll hand this over to David.
Well, hello, everybody. I'd like to introduce the concept of the genetics of PKD today, and I can start off with the father of genetics, a name probably many of you know, Gregor Mendel. He was actually born Johann, but like some students today, he could get a free education if he became a friar, and the friars gave him the name Gregor, but he was born Johann. And he was fond of peas. And before I get into the story of Mendel's peas, I wanted to put up a quote by another famous scientist, Albert Einstein. And it's, if you can't explain it simply, you don't understand it well enough. And I want to apologize in advance in that if I don't sound simple and straightforward, it may well be that I don't understand it well enough. It's been quite a while since I've taken genetic courses. And genetics has progressed a long way, not only since Mendel, but since Watson and Crick. So please bear with me as I try to explain the genetics of PKD. So Mendel's peas. What he did, and what he was curious about, was why when he bred a yellow round pea with a wrinkled green pea, that in the first generation you always got a yellow round pea. And what he realized is that when he took those peas and bred them with each other, and you had the next generation, you got a whole range of different types of peas. Now the majority, as you see, are yellow, and the majority are round. And this really explains what I'll be getting to later in the talk about why you really have two forms of polycystic kidney disease, the autosomal dominant form and the autosomal recessive form. So as I hope will be pretty obvious even by this first slide, is that yellow and smooth are dominant and green and wrinkled are recessive. And that's very simply put in this diagram by capital R's for round and capital Y's for yellow as the dominant gene, and the uncapitalized R and uncapitalized Y for the recessive gene. So dominant obviously dominates over the recessive traits, and we'll be getting back to this as we proceed. What wasn't known in Mendel's era was how these traits were transmitted from one generation to the next. And what I'm showing you is one of many slides you can find on the web of the DNA double helix, which was discovered by Watson and Crick and Rosalind Franklin in 1953. And this really opened up genetics, because it's this structure, and I'll get into discussing this structure a bit more, that contains the code to who we are and to who all animal species are. Our DNA is unique, and it's unique between species, and it's unique within species. So the structure of DNA, as I mentioned, was figured out in 1953, and that double helix was critical because it allowed other scientists to understand that if you opened up that double helix and looked at the single half of that double helix, it was covered in compounds that are called bases, and these form base pairs, and the sequence of these base pairs is the genetic code, and I'll show you that in a couple of slides. So the genetic code for how DNA encodes for making proteins was deciphered, and this was brilliant work that was done in the late 50s, early 60s, and 70s. And one of my professors at the University of Chicago was actually the one who showed, by techniques that aren't even used today, that all cells in the body that contain a nucleus, so that excludes things like red blood cells and platelets, contain exactly the same DNA. Before that experiment, people thought that a muscle cell must contain different DNA than a cell in your stomach, for example. But it's now known, and it's been known for some time, that all cells contain the same DNA. So the organization of these genes are still being figured out, and the regulation of these genes is still being figured out. Regulation is important because whether a gene is expressed or not determines whether it's going to be a muscle cell or a stomach cell. And it also goes on to explain how mutations support the theory of evolution even, because mutations occur randomly. Everybody has them. They occur relatively infrequently, but sometimes they don't really do anything and they're carried on to the next generations. And this is one of the ways that organisms, including humans, slowly evolve. Now, the other beauty in figuring out DNA is it was a spot-on explanation for Mendel's P's and those classic experiments in the 19th century. So we talk about autosomal dominant PKD and we talk about autosomal recessive PKD. So dominant traits require only one copy of the DNA from one of the parents. These copies are called alleles. And if you have one parent who has autosomal dominant PKD, then if the child inherits that gene will dominate over the normal gene inherited from the other parent. On the other hand, recessive traits require two copies, one from each parent, for the expression of that gene. And I want to introduce a word for expression that's used in genetics. The genotype is the code that we all have that is the instructions for building a human being, and in particular for building you individually as a human being. And the phenotype is how that code is expressed. So, for example, if you have genes that say that allow you the capacity to become 6 feet tall, if you have a lousy diet when you're growing up, you're likely to be 5'8" instead of 6 feet tall. So although you have the coding to be 6 feet, your genotype says 6 feet, your phenotype can be variable depending on your environment, other diseases you may have, your diet, and so forth. So those are two terms that are useful in genetics, genotype and phenotype. So ADPKD, as most of you know, is the most common form of PKD. As a matter of fact, it's one of the most common autosomal dominant genetic diseases that there is. It's definitely the most common renal genetic disease. And it's due to a mutation in one of two genes, the PKD1 gene or the PKD2 gene. These are both genes that are codes for proteins called polycystin 1 or polycystin 2. And 85% of people with ADPKD have a mutation in the PKD1 gene. About 15% have a mutation in the PKD2 gene, which is has a more mild phenotype. In other words, people with PKD2 mutations tend not to go into renal failure as soon as people with the PKD1 gene. And as I said before, one parent has autosomal dominant polycystic kidney disease, and that means, because it's a dominant trait, that there's a 50% chance that each child born to such a couple will inherit autosomal dominant polycystic kidney disease. Now, in contrast, ARPKD, or autosomal recessive PKD, is a very rare form of this disease. It's caused by a mutation in the fibrocystin gene, or the Ph.D1 gene. If you'll notice, I'm following the format that is used when you write down a gene, that you put it in italics, whereas the protein is not put in italics. So in the case of autosomal recessive PKD, both parents have the gene, but neither parent has the disease because it's recessive. So in that case, just like Mendel's peas, where you saw that the majority were yellow and smooth, there's only a 25% chance that each child of a couple who each have an autosomal recessive PKD gene will actually have autosomal recessive PKD. Now, I'll be talking a little bit later about the statistics and probability around this. These are not exact numbers. As a matter of fact, some people think Mendel did a little cheating on counting up his P's to get at the 25%, because if you do the experiment multiple times, it'll come out 27%, 22%. It's like flipping a coin, basically. So what does autosomal dominant polycystic kidney disease look like? And so here are two parents. One is affected, darker teal. One is unaffected, lighter teal. And these objects, which I'll explain a bit more, are chromosomes. We each have a pair of chromosomes, one from our parent and the other parent. And when you look at the inheritance, you'll find that 50%, even though they have the unaffected gene, will not show the disease at all. But if you have one of the genes from one of the parents, you'll see that you'll be affected. So this is the 50-50 chance I was talking about. So if you have two of the lighter teal genes, you're unaffected. If you have one of the affected genes, you'll have the disease. So the phenotype here is dominant. When you look at this, from an autosomal recessive polycystic kidney disease perspective, you see that both parents have the recessive gene, but because it's recessive, neither parent has the disease. And if you look at the possible combinations of these chromosomes, you'll find that two of the children in this illustration will have the gene for autosomal recessive PKD, but will not have the disease. But they're carriers because they'll still have one gene for ARPKD. There'll be one child that has no gene for autosomal recessive PKD and therefore is not a carrier, and her progeny will not have the disease. On the other hand, 1/4 of the children born to this hypothetical family will have both recessive genes from each parent, so the lighter teal here. So 25% of the children in this hypothetical family of 4 children, you'll have one child with a disease, 2 carriers, and one child who no longer has the gene for ARPKD. So now about the statistics and probability. Many of you, and I know I've had the same conversation with my own family, is that the chance of ARPKD or ADPKD of each child in a PKD family is independent of the other child. It's like flipping a coin once and then flipping it again. That second flip will have no relationship to the first. So there are some families where all the children may have PKD. There are families in which no children may have PKD. And so if you think of that hypothetical family of 4 children, some will have 4 boys, no girls, some 4 girls, no boys, so on and so forth. And so it is with PKD, whether it be AR or the AD form. So with recessive inheritance, the odds of having 4 children without PKD are greater than for ADPKD. So there are other modes of inheritance. One I think is important to discuss, that's spontaneous mutations. Now, if that spontaneous mutation in the PKD1 or 2 gene occurs in the egg or the sperm from the parent, in other words, the mutation occurs not as a result of inheritance for the parent, but occurs randomly in the sperm or the egg, because as I mentioned, evolution in part is sparked by these random mutations, and some of them are bound to occur in the PKD1, PKD2, or fibrocystin gene. This can lead to spontaneous autosomal dominant PKD, because it's very unlikely that you will have 2 mutations in the fibrocystin gene. If mutations occur somatically, and by somatic I mean not in the germline, in other words, not in the egg or the sperm that forms the new human being, these are mutations that often can cause cancer or other benign things like moles or so forth. There's also something called epigenetic inheritance. Epigenetic is actually a very simple word, and it means extragenetic. So DNA can be modified chemically, and that can be inherited as well, and that can affect not the genotype, in other words, whether you have the mutation or not, but it can express the phenotype. So some epigenetic inheritance can cause a more severe form of disease or a more benign form of disease. There is some DNA in mitochondria. I'm not going to go into that. And as I'll show you in a bit on what chromosomes look like, there are sex-linked inheritance patterns, but that's really not pertinent to PKD. So where is most of the DNA in the cell? Well, it's in the nucleus, and I picked this fuzzy picture because it happens to contain a cilium, which is where the products of PKD1 and PKD2 and fibrocystin are located. And within the nucleus are chromosomes that are not obvious when the cell is not dividing, but when they divide, the DNA becomes organized into these chromosomes. And these chromosomes contain the double helix that I mentioned that provided the answer to that question of why Mendel's peas did what they did and how inheritance happens. So looking a little more closely at this double helix, you'll see that it's really formed by ladders, and that the yellow backbone is really sugars, and they hold these base pairs together. The names of these base pairs, there's only 4 of them, is adenine, thymine, guanine, and cytosine. And A binds to T, and G binds to C. And for those of you who are science fiction fans, you might remember a movie called Gattaca, and actually Gattaca was a sequence of code in DNA. So G binds to C, A to T, T to A, T to A to T, and so forth. And it's this sequence of these 4 base pairs that contains the entire coding for what we are as individuals and human beings. So I mentioned that the genes are in chromosomes. In humans, there are 23, and males are a little short-shrifted. They have an X chromosome and a Y chromosome. The Y chromosome, as you see, is much smaller and therefore contains less genetic information, whereas females contain 2 copies of the X chromosome. And that's where some of the X-linked or sex-linked genetic conditions come from. But for our purposes, it's 23 chromosomes, and I'll show you, just for the sake of curiosity, where the genes for PKD are located. But just to summarize, that what does it take to code a human being? Well, it's 3 times 10 to the 9th base pairs of those nucleotides, A, T, G, and C. How much is 10 to the 9th? Well, if I'm not mistaken, that's about 3 billion base pairs. So our instructions for making a human being are about 3 billion base pairs. And that's organized into 22 autosomal chromosome pairs. And this will give you a better idea of what autosomal actually means. These are the chromosomes that are not sex chromosomes, such as X and Y. So both females and males have 23 chromosomes each. They're paired, so you have 46 in total, but you have a copy of one from your father and one from your mother. And the males, as I mentioned, have XY, females have XX. So where are they located? Well, the PKD1 gene is located on the short arm of chromosome 16. I'm sure this doesn't mean a lot to many of you. To be honest, it doesn't mean a lot to me. But this is chromosome 16, and these banding structures are something you can see under the microscope, and the molecular biologists and geneticists, who are much smarter than I, have figured out where genes are located on each of the chromosomes. And oddly enough, PKD2 gene, or polycystin 2, is located on a completely different chromosome 4. Yet these 2 proteins, one from chromosome 4, one from chromosome 16, are coded for on completely different chromosomes, even though polycystin 1 and polycystin 2 act together in the base of the cilium. Whereas fibrocystin, or PKD1, that's on yet another chromosome. This, as you remember, is the gene that is responsible for autosomal recessive PKD, and it happens to be located on chromosome 6. So this is a very complicated slide, and it's just to show you how complicated genes really are. So here's the gene in black, and you see that it's not just a series of coding of those base pairs, but it contains areas that don't code for anything. Those are called introns, and then there are also exons that actually code for the protein. And this is a highly schematized look at a linear cartoon of what polycystin 1 looks like. And if you look at all the arrows, it shows you where all the mutations might occur. And these mutations may be as simple as changing a base pair, that you have A and T, and it may become G and T, and that doesn't even form a good base pair. That's a simple mutation. There are also, and then also can be called a nonsense mutation. There are missense mutations that cause the wrong amino acid, which is how what proteins are made of, to be inserted. There are insertion mutations where actually extra DNA is inserted. And there are deletion mutations where, in some cases, large portions of the sequence in the DNA are actually missing. And one of the things that are being done actively at the Mayo Clinic and elsewhere, and the reason why the MODIFY trial is so important, is to try to find out how these very different mutations which all end up resulting in cysts in your kidneys, operate? And are they related to the severity of your disease, the time of onset of your disease, and so forth? So for those of you who may have contributed a blood sample to the modified trial, this is what's being done with it. My family and other families are very unlikely to share the same mutation. About 2% of any given mutation that's been discovered in PKD is shared among families. So it means that if you meet another family that has PKD, you're very unlikely to have the same mutation as that other family. So what happens with this sequence? So here's that double helix. It gets opened up, and another word, transcription, is the DNA code is read, and it's turned into RNA, and the exons, which make the protein, are all spliced together. This is a very complicated process, which would be the basis of several other webinars if people were interested. But let's just say that this mRNA, which is called messenger RNA, leaves the nucleus, and it gets processed in something called a ribosome. And a ribosome is made up of proteins and RNA, and it's really the factory that turns the message that came from the DNA into the protein. So you can see the message being read like a ticker tape going through the ribosome, and the amino acids, which are these little blue structures, are added according to the genetic code to this growing polypeptide, which will become the protein that this messenger RNA has coded for. So that's a couple minutes discussion of what is really elegant science that has taken decades to understand and is still being understood. But I just wanted to give you a feel for what this all looks like. And maybe in the future I will talk a little more specifically about what's going on in this slide. So I showed you a linear sequence of polycystin 1. Here's both polycystin 1 and polycystin 2, and what they look like, again, it's schematized, but it gives you a more two-dimensional feel for what's going on. And here's the outside of the cell, and polycystin 1 is a very interesting protein in that a lot of the protein is actually outside the cell surface. Here's the cell surface, and here's a good portion of the polycystin 1 protein. Just to give you an idea of scale, this is the cell membrane, which is about 70 nanometers thick. That's, to give you an idea, a nanometer is 10 to the minus 9th meters. So it's very tiny. This is almost atomic in size. And the interesting thing, even though they come from different chromosomes, is here's polycystin 2, a smaller protein, but it's right adjacent to polycystin 1, and very curiously, they both sort of tangle together inside the cell. So outside the cell, inside the cell. And you can see that polycystin 2, although it has these portions, these blue cylinders that are in the membrane, it doesn't really have as much going on the outside as polycystin 1. Whether that has anything to do with why polycystin 2 mutations are less severe than polycystin 1, I have no idea, but I think this is a good place where research should be happening, because this is where the mutations occur, and this is a critical interaction that is still poorly understood. So getting back to the genetics, genotype leads to the phenotype. So the phenotype are your observable traits, and are the result of gene expression. So eye color, whether you have red or brown hair, and so forth, are the phenotypic traits. And these traits, just like Mendel's P's, may be dominant or recessive. So you may have one gene for red hair, but if you have another gene for brown hair, you'll have brown hair. And actually, hair color turns out to be a little more complicated than that. But the type and location of the mutation within a gene can affect the severity of the resulting phenotype, as I was trying to describe earlier. So some of the big truncation mutations, where a lot of the sequence for the protein is missing, may lead to more severe disease. And there are other modifiers, and that's why the ongoing genetic study is called the MODIFY study. And the other allele, in other words, you get one allele or one copy of the gene from your father and one from your mother. It may be that the gene from the unaffected parent has some activity relating to the gene from the other parent. There's also an active search going on for modifier genes, hence the name MODIFY trial, and that's under active investigation. In other words, are there genes we don't even know about that affect the phenotype of polycystic kidney disease? And of course, there are environmental effects like your diet, injuries that you may have had, sports injuries in particular, other diseases that you may have. These may all affect the phenotype or the expression of the disease, but not the genotype, which is the coding for that mutated protein. So some mutations may have no effect or even a positive effect. One example is sickle cell anemia. If you only have one gene from a parent and not two, you actually have resistance to malaria, and that resistance is positively selected for during evolution. Unfortunately, I don't know of any benefit of the PKD mutation, but it does occur typically later in life when people are already of reproductive age, and therefore it can be inherited without even knowing that you have it at the time you have children. And as I mentioned, not all genetics is strictly Mendelian. I mentioned epigenetics. These modifications of DNA and the proteins that hold DNA together may be heritable. So it's not the coding that's inherited, but the modifications of the genes that are inherited. And these chemical modifications affect how genes are transcribed. Some of these modifications turn off a gene, or others turn on a gene. And again, that might be another topic of discussion. And this has to do a lot with why muscle cells are muscle cells and stomach cells are stomach cells. And then I mentioned RNA, and DNA clearly doesn't just code for proteins, because it's actually a copy of DNA made out of RNA that is the messenger for that code for proteins. But there are a lot of RNAs other than messenger RNAs. There are rRNAs, or ribosomal RNAs, that make up the machine that in the cell that makes proteins. And then there are sequences of DNA of unknown significance. We used to think, or not me, but geneticists used to think that some of this may just be garbage, but increasingly we're understanding that it's not garbage, and it actually codes for small nuclear RNAs, small nucleolar RNAs. I'm not going to get into what these are exactly, but there are also a class of RNAs called regulatory RNAs, and in particular the microRNAs, here expressed as miRNA. In the recent literature and at the American Society of Nephrology meeting that was just held, it's clear that these may hold the key to therapeutic approaches to polycystic kidney disease that are very exciting. So the reason they're exciting is that these microRNAs, which may only be 17 to 21 bases long, in other words, they're very tiny, they don't code for proteins, but they can affect cell proliferation, which is an issue in PKD, affect cell polarity, in other words, which side is up and which side is down, another issue in PKD. They can modify cell secretion. Obviously, secretion is a big issue in PKD because that's what fills the cysts. And it also can cause cells to die in an orderly process called apoptosis. The second P is silent. And this is very important because if we could get cells of the cyst that line the cyst to die in an orderly fashion, the cyst would not grow as quickly, or maybe not grow any further. So this is an important area that I'm watching and I'm excited about. And there is other work, it's only in rodents right now, but that kidney cells with normal PKD1 or PKD genes may actually be transplanted into rodents that have the human rodent equivalent of PKD, and they get restored kidney function. So there's a lot going on that are not your typical drugs. In other words, they're not small molecule type drugs, but they may be a form of RNA, or they may actually be a cell transplant. There's a lot of things going on that I think are leading to a renaissance in PKD as compared to what was known when the foundation was first formed in 1982. So to sum up, ADPKD is one of the most common, if not the most common, autosomal dominant genetic disease. And ARPKD is much rarer, as are most autosomal recessive genetic diseases such as cystic fibrosis or ALS and so forth. And nobody's DNA is perfect. We all carry mutations. Most of these mutations are silent. We don't know we have them. And we also carry bits of DNA from viruses that are actually inherited. And as I mentioned, there's DNA with no known function that was called garbage DNA, but perhaps it does have function, and it'll be exciting to find out what that function is. So many unanswered questions remain, but I hope with the discussion of science I haven't obscured the main point of this talk, and that is how ARPKD and ADPKD are inherited, and why some families may have no children with the disease and some families may have all children with the disease. And with that, I'd be happy to answer any questions.
Okay, David, great. So we do have several questions, and the first one is, can PKD skip a generation?
I have not heard of that being the case unless it was perhaps a PKD2 mutation that was so mild that one generation was never aware that they even had the disease. Other than that, I'm not aware of a genetic mechanism for skipping generations.
And so in that case, it wouldn't technically be skipping a generation, it just wouldn't be noticed as something that actually happened, but it was actually there.
So that is a good example of genotype versus phenotype. That in some cases, although you have the mutation, the phenotypic expression of that mutation is so mild that you never know that you even have the disease.
Okay. Okay, I'm not sure if you'll be able to answer this or not, but if you can, then just say that, and we'll find someone who can and put it up on the website later. Is there genetic testing to see if you have a mutation in the PKD1 or PKD2 gene, and if so, would knowing this information be helpful to doctors and patients?
There is the means of testing for the genetic mutations for PKD. Their accuracy is not 100%. They're very expensive. I think there is some value in knowing whether you have PKD1 or PKD2, but I'm only speaking personally. The fact of the matter is that treatment in the United States, at least, is according to your symptoms, and it will be clear whether you've been genetically typed or not, whether you have a less severe or more severe form of the disease, and you'll be treated accordingly.
Very good. Very good answer. Okay. So, okay, this is an interesting one. If a patient has had PKD for 30 plus years and has not needed dialysis and is told to be in stage 3 chronic kidney disease, should one assume that the patient has PKD1 or PKD2 gene? Is there any way to make an assumption based on your phenotype about your genotype?
That's a toughie, and I would defer to a nephrologist and a nephrologist familiar with genetics for that question. But it's fortunate that one has been able to go 30 plus years and not have end-stage renal disease.
Okay, so I think this kind of touches on what you said a second ago about genetic testing not being 100%, not 100% accurate, but this says that her daughter has PKD by ultrasound, so I'm assuming they've been diagnosed by ultrasound but not by genetic test. And then her mother, her aunt, and other relatives also have PKD. The daughter was genetically tested, but the test came back negative. They could not find a gene for PKD. How could this be?
Well, as I mentioned, to my knowledge, and again, I'm not speaking definitively here, that because of the complexity of the polycystin 1 gene, or the PKD1, a definitive genotype cannot be obtained in every instance, and I think it has about 85% accuracy. I also know, but I can't explain why this is important, you would need to talk to a geneticist, that having other family members tested can help make a more accurate genetic diagnosis in the case of PKD. So, but that becomes prohibitively expensive since the genetic testing, as I understand it, for PKD is around $2,500 to $3,000 per person. So if you were to test 4 or 5 people or more, it becomes quite expensive.
Okay, so this person is saying they've heard of ADPKD spontaneous mutations. Are there any cases, to your knowledge, where this occurs with ARPKD? Maybe one parent is a carrier but not the other, or neither parent is a carrier, and ARPKD is the result of a spontaneous mutation?
I personally am not aware of that. I have the feeling that because it is so rare that there may not be enough information. Again, a pediatric nephrologist with a genetics background is the person to ask, and perhaps we can forward that question to someone on our scientific advisory committee who might be knowledgeable about that question. But good question.
Yeah, we absolutely can, and we will do our best to get some information about that. Okay, so this is a good question. Someone is asking about tests for PKD and also using PGD IVF, which is pre-implantation genetic diagnosis, and using in vitro fertilization. And I'll let David comment, but I will say that we have an entire webinar on PGD and IVF logged on our website, archived there, so please check that out when you get a chance. But David, would you like to comment on that here?
I'll comment briefly, and I would, as Alexis says, go to the webinar because it was given by somebody very knowledgeable in this. But when the embryo is at a stage of 16 to 32 to 64 cells, or maybe even 8 cells, I'm not sure, one of those cells can be taken from the embryo without ill effect, or at least with minimal ill effect, and actually that one cell can be diagnosed, if you will, for whether it has the PKD mutation or not. And if not, then that embryo, minus that one cell, can be implanted, and that is the basis of that particular technology.
Great. And I think that person also was asking about the specific genetic test for PKD, but I think, as you mentioned, you don't have a lot of in-depth knowledge about that. But we do have a bit on our website, and I can highlight that a little bit more in the same area as the webinar, so you can check out our website, and I'll try to add in some information about genetic testing specifically for your ease of access. Okay. If you have no symptoms of PKD, but one of your parents has it and you are checked in your 40s, can you be checked in any way other than a sonogram to see if you have the disease?
Well, so your ultrasound at the age of 40, I think, is pretty definitive. More expensive, you can, instead of ultrasound, use MRI or CT scans, but ultrasound is, the most inexpensive, and CT scans involve X-rays, so and MRI is expensive, so I think ultrasound is probably pretty definitive by the age of 40.
Okay. Just a few more questions. So this says, my father, the oldest of 10 children, was the only child that had PKD. Is this rare? I'm an only child. Or this person is the only child that has PKD, and their 3 children now in their 40s all have PKD. Did you follow that?
I think that was covered in my slide about probability, and I use when you hear of a family that has 10 boys in a row or 10 girls in a row, or even more, 14 of the same sex. The same analogy applies to PKD, that although on average, if you have one parent with autosomal dominant PKD, 50% of the offspring will have the disease. It is a possibility, and it does happen, that none of the children will have PKD, or all of the children will have PKD. It's a matter of probability.
So when they're asking, is it rare for 1 of 10 to have PKD, you would say yes.
Extremely. Yeah. It's just like, how many times can you flip a coin and get heads 9 times in a row? Unlikely, but possible.
Okay, this one's kind of in depth, so here we go. My brother and myself both have ADPKD, but my GFR is stable and my brother's is not. Is it possible that he got the PKD1 gene and I got the PKD2 gene? So let's start there, and then there's a second half.
I think unlikely, because one parent would have the disease, and that person would either have the PKD1 gene or the PKD2 gene. A question I can't answer is if there are people with mutations in both genes. Again, we'd have to refer that to a geneticist.
And I will say I did ask that question once and was told that no. I of a geneticist, and they said no, that would not be compatible with life. Okay, so then would that be kind of an example of your genotype and your phenotype? You know, you can have the same genotype but a different phenotype.
Exactly.
Okay.
And men and women often have in the same family different ages of onset of PKD, or different levels of severity of PKD. In other words, some people, even with the PKD1 mutation, may never need renal replacement therapy, and others in the family might.
Okay, so now the second half of this question. This person has a son, and they're saying they have a son with ARPKD. Is that rare from inheriting a PKD gene from her and the other gene from her spouse? So I guess really the root of this question is it, I'm not sure it's possible if you have ADPKD for your child to have ARPKD, or maybe it is and I'm misunderstanding.
No, I don't think that would be the case. I think the geneticist who told you he's not aware or she's not aware of someone with both a PKD1 and a PKD2 mutation, because ARPKD is recessive, each parent has a mutation in the fibrocystin gene.
Now, there are cases of ADPKD onsetting earlier in life. So although it's not ARPKD, it might onset at an age that might lend a physician who's not familiar with genetics to think that that's what it is.
So AD in older literature, sometimes I think the abbreviation for the disease would include the word adult, but it's very clear that ADPKD, while it's predominantly first becomes issue in adults, does happen in children. This is a more severe form, but it's still autosomal dominant PKD, and pediatric nephrologists who are familiar with the disease do have patients that they treat who are children, unfortunately, but it does happen.
Okay. So how common is spontaneous mutation of the PKD gene? I'm the only one in my family. Both parents were without kidney cysts in their 80s.
So I'd say if your parents were in their 80s without cysts, that they did not have PKD. I think that about 10% of cases of ADPKD are not accounted for genetically. And I'm not sure if that's because of the uncertainty of the genetic diagnosis. In other words, I mentioned that not all genetic tests will show the disease even though you have the mutation. But so that 10% figure is plus or minus, but it would appear that a significant percent, although small, are spontaneous cases of the disease.
All right. Well, that's all the questions that we have. I'm so excited. We hardly ever get through all of the questions, so it's nice to do that. And we are nearing the end of our time anyway, so we are just going to wrap this up. David, can you advance to the next slide? I think that it has our...
Yes.
So our upcoming education events. So we have an evening webinar series coming up on December 2nd at 7 p.m. Where David will discuss the sequence of drug development and answer the question, why is it so darn hard to find a therapy for PKD? So this is really in David's wheelhouse. He's got a lot of experience with this, and we are really excited to hear what's taken so long and what are we doing to move towards that direction. Also, be sure to follow the PKD Connection blog. It's it's a place for patient stories, education posts, and more. You can find that at pkdconnection.org, and you can sign up for notifications every time we put up a new blog. And then we also have the PKD National Convention coming up next year in Orlando, June 24th through 26th, 2016. You can check out pkdcure.org/convention for updates. And I do want to give a quick note that next month, December, we will not have a webinar Wednesday because we have the evening webinar on December 2nd, and because it is the holiday season, we thought we'd take a break, but we will be back in January with fresh topics and ready to go for a year of webinar Wednesdays. So thank you so much, David, for all of this wonderful information, and thank you to the audience for participating and submitting your questions. If you want to reach out to us directly, you can email us at education@pkdcure.org, and a member of our education team will get back to you just as soon as we can. On behalf of everyone at the PKD Foundation, thank you for joining us today, and have a great afternoon. Bye.